Okay, thank you very much. So today I will present you some of the experiments that we developed in Marseille to understand the molecular diversity that can be found during the formation of planetary system and mainly by focusing on solar system. So when we are interested in to obtain information of this molecular diversity, we can have direct information by analyzing on Earth meteorites, because you know that meteorites have asteroids with parent bodies. And so some of these meteorites, so not all meteorites, but you have carbonaceous conduit meteorites that present a fraction of organics in their content, so up to 5% of their weight. And this these organic molecules are divided in soluble fraction and insoluble fraction. So we can analyze these different fractions to obtain information about their molecular content. So for instance, you have Philip Smith Copley in 2010 that developed a new approach to analyze these organic contents using high-resolution mass spectrometry. So which is interesting with this technique is that you have the enough resolution to separate isobars that give you direct information about the number of molecules that form that sort of organic contents. And as you can see also, because we have the high resolutions, we can obtain the atomic composition of each ion. So we can know if you have carbon, hydrogen, oxygen, nitrogen, sulfur in the system. And what you observe is that you have 10,000 different molecules that form this organic content. So you have a very important molecular diversity that are present in these objects and that are maybe related to the origin of the solar system. So you can also perform targeted analysis. So for instance, often they focus on some what we could say biomolecule precursors, so sugar. So they observe sugar, nucleobases, and various amino acids, up to 90 different amino acids, for instance, in the Murchison meteorites. So that shows you that in the soluble organic fraction, you have an important molecular diversity that were observed. So as I told you before, you have also an insoluble organic fractions. So it's more related to macromolecules that are related to aromatic cycles that are linked with aliphatic bridge. And so now the question is that where come from these organics? So you have different scenarios. So the ones that we developed in the lab in Marseille come from the dense molecular cloud ices, and we will simulate in the lab their evolution during the formation of solar nebula up to the formation of the protoplanetary disk. And then we try to, so here we will see what sort of molecules can be generated in the gas phase, in the solid phase. And then we try to understand what sort of evolution could occur inside the parent body of meteorites that are asteroids. So the first step is to develop experiments that will simulate this first evolution, what we call pre-accretional evolution. So what's that? We will form in the lab analogs of ices as they are observed in dense molecular cloud ices, and we will submit them to different energy processing that they can undergo during the solar nebula evolutions. And we will try to understand what sort of organisms can be released in the gas phase during the warming process, and at the end, what sort of organisms can stack on the surface of the grain. So for that, we develop different experimental setups. So here you have the ones that we developed with Fabrice Duvernay, which is called AYA. So the center of the system is this vacuum chamber. And after, we have different analytical tools, so infrared spectroscopy, which is the basic experiments that we use for the analysis of ices. And we have, for instance, here gas chromatography coupled to mass spectrometry. And I will present you what sort of information we can obtain with that sort of system. So the first experiment that I will show you is that we start from very simple ices, including water, methanol, and ammonia at 20 Kelvin under 10⁻8 millibars. So here I will only show you experiments results that we obtain with UV radiation. We perform so with ion and electron, but I'll only focus on UV radiation. So we irradiate at Lyman alpha during 24 hours this ice, and after we will warm the ice and we will recover all the molecules that can be released in the gas phase during this heating step. So here you have another representation of the experiments. So at 20 Kelvin, we condense the ice, we irradiate it with UV. We can have an in situ information about the ice evolution with infrared spectroscopy, and after we warm the system, and we will recover the organic compounds, the volatile organic compounds, to inject them in the gas chromatography. So the gas chromatography will allow you to separate the molecules before their entry in the mass spectrometer, excuse me, here, to obtain their identification. So when you start from this water, methanol, and ammonia ice that you submit to UV radiation, that you warm this ice after processing, we observe the formation of more than 40 different molecules. So here, these are only the molecules that we were able to identify. You have much more signals that were not identified yet. But as you can see, by starting only from these 3 molecules, you can generate and observe in the gas phase a very interesting molecular diversity. You have alcohol, ethers, aldehydes, you have ketones, you have also nitrogen compounds, CHO compounds, and you have ester acids and D-ethers and alcohol ethers, that sort of thing. So this very simple result show you that by only starting from 3 very simple molecules at low temperature, but if you put energy to the system, you are able to form an interesting molecular diversity that is observable in the gas phase. So this solid phase, solid ices, can directly generate an interesting molecular diversity that you can observe in the gas phase. So where come from these molecules? So the main part of these molecules come from the radical chemistry. So here you have the different initial molecules, water, methanol, ammonia. So I will not detail you here the different branching ratio, but which is interesting is that if you modify the ratio in the initial ice, you modify this branching ratio and you directly can have insight of this modification in the gas phase. So I will not detail here how this modification impacts the gas phase, but if you want to have more information, you can go to the poster of Laurie Arnold, and he will present you how the modification of the initial ice can impact the gas phase and how the gas phase can be used to obtain information about the chemistry that occur in the solid phase. Okay, so from this experiment, we've seen that in ices, these ices present a very interesting chemical reactivity when you submit this ice to energetic process. You have radical chemistry, but also when you warm your system, you will be able to obtain thermal reactivity. And so, of course, at a point, the water will desorb, so will drive a large part of the molecule in the gas phase, as we observed in the previous experiments. But which is also interesting is that the water have an important effect because it will allow molecules to stay in the matrix. The water matrix are higher temperatures than their own desorption. And that allows to obtain a very interesting thermal reactivity that will lead to the formation of more refractory molecules that will lead to the formation of what we call refractory organic residues that will stack at the surface of grains and in our experiment of sample holder. So now we will try to understand what sort of organisms can be generated during the processing of these ices and that stack at the surface of the grain. So classic analyses are based on infrared spectrometry analysis. So here you have the results that were obtained with Louis de Cour at the Institut d'Astrophysico-Spatiale with the MICMOC experiment. So here you have a microscope picture of the residue. So in fact, this is an organic oil that you obtain. Here you have infrared spectroscopy. So you can identify classical molecules that are obtained in this residue, hexamethylene, tetramine, excuse me, polyoxymethylene. You obtain with infrared spectroscopy, excuse me, some interesting information about the chemical functions that form these different molecules. But we are still limited with this technique to obtain information about the molecular diversity that can be generated. And the question is that, as you've seen, we can generate molecular diversity in the gas phase, and is it the same for the solid phase that we form at the end of the experiments? So to obtain this information, we develop a new strategy with Roland Thyssen and Véronique Hueton at the IPACT. So in fact, we analyze with high resolution mass spectroscopy this residue. So we recover the residue with methanol, is entirely soluble in the methanol. And as you can see, at the end, you have a mass spectrum that presents thousands of different molecular ions. So the first step was to verify that these molecules that we observe come from the ice. In fact, we demonstrate that 95% of the ions that you observe here come directly from the methanol that were present initially in the ice. So we demonstrate that when you start from three very simple molecules, water, methanol, ammonia, at very low temperature, submit again energy, UV, you have the same for with ion and electrons. You warm the system, you have the volatiles that go to the gas phase, and you recover at the end an organic fraction that present thousands of different molecules with masses up to 4,000 Datsun once you start from only methanol as a source of carbon. So that shows you that in this environment, you have very complex and rich reactivities that occur and that will lead to a very important molecular diversity that present, in fact, similarities to what is observed in meteorites. So you can also perform targeted analysis as was performed on meteorites. And we observe nucleobases, you have sugar, you have amino acids, and you have also small peptides. So that show you if you compare what we form in the lab from ices and what is present in the meteorites for the soluble part, you have some similarities, excuse me, similarities, yes. So we form this soluble organic matter. That was the question, is it possible to form the insoluble organic matter from these ices? So we perform a very simple experiment. So here you have a new representation of the same data that I presented to you before. So because of the high resolution mass spectrometry that I told you, we can obtain the stoichiometric formula for each ions. And because we have this number of carbon, hydrogen, oxygen, and nitrogen for each ions, we can obtain the elementary ratio He and OC. So this is, which is important here, is to only look to the evolution of the distribution of the ion in the space here, okay? So we start from the soluble fraction that was formed from these ices, water, methanol, and ammonia. Here you have the top of the residue before the irradiation. So what we performed to form this insoluble fraction, we submit this soluble residue to UV irradiation. So you have here before the irradiation and after the irradiation. So you can clearly see that we modify the surface of the residue. And if you compare before and after the irradiation, you can clearly see that we transform our initial organics that were soluble. And we verify by washing with water that this fraction is totally insoluble. So that means that starting from our ice, we can generate soluble fraction and insoluble fraction. Oh, 3 minutes. So of course, this is materials that we form in the lab. So now which is important is to compare, to confront what we form in the lab to what is observed in meteorite. So we apply exactly the same analytical techniques to compare what was formed from this IC to what is observed in meteorites. So if you look to the insoluble organic matter, you can see that we have quite the same molecular distribution. Even if here also, I will not say this is exactly the same organics that we form, but we are able to form insoluble fractions that present similarities. But which is important here is that if you look to the soluble organic matter, what observed in Paris meteorites is totally different from what is observed from what we form from these ices. So that means that the soluble fractions that we form from these ices in the lab are totally different from what is observed in meteorites. And this can be explained by the fact that here I only show you the first step that start from the ice, and we are on the protoplanetary disk where we can observe soluble organic matter and maybe insoluble organic matter. But if you look to the meteorites and mainly on the mineral phase of the meteorites, we know that you have secondary alteration in asteroids and mainly linked to chaos alteration. So the question is that in these bodies, you can have a post-accretional evolution that lead to the transformation of the initial organic matter. And to verify that, we perform very simple experiments. So we took the residues that come from the ice processing. We put them in simulate asteroidal conditions, and we look to what happened. And so you have at the left before and at the right after the acceleration. So you can see that we have a transformation of the molecules. So here we start from 10 of 1,000 different molecules who have the same molecular diversity after the alteration. But you can see that we have a transformation. We lose oxygen and we decrease in He ratio. So the question here again was, okay, when we perform this alteration, aqueous alteration, what happened and how we can compare that to what is observed in meteorites? So here I will put you in green. I superimpose what is observed from soluble organic matter of Murchison meteorites. So again, from the residues that directly come from ice, clearly you have a huge difference. But if you look after the aqueous alteration, you see that I will not say this is the same, of course, but you see that we transform our initial organics with higher similarities to what is observed in meteorites. So which is interesting here is that we can present a scenario that start from the solid phase of dense molecular clouds, so from these small icy grains, and then when you submit them to different energetic processes, you can have different steps of evolution. You can form soluble and insoluble organic matter that maybe can be present at the surface of grains around the protostar, and then during the after the accretional steps, you can also have a secondary evolution that leads to a new evolution inside parent bodies of meteorites. And this is one of the scenarios that could explain what you observe in different meteorites. So thank you for your attention, and thank you for all collaboration and funding that allowed to this work.